Thank you very much, Angita and Liang, and it's really a great pleasure to be speaking here today. So thanks so much for the invitation. It's a pity I cannot be there in person. I quite miss the Boston area. So yeah, but anyway. So yeah, so today I will be describing our recent work on basically the algorithm that we've developed called somatic, which aims to detect somatic mutations in single cell profiling datasets. Let's see. Yeah. So before getting into that, so just to briefly describe that in my group, we have three main areas of research. One is related to understanding the mechanisms and consequences of genomic instability processes. What I will be discussing today, which is really understanding what is the effect of somatic mutations on cell phenotypes, and that relates to cancer as well as what is the impact of mutations in differentiation and in normal cell behavior. And finally, we're also very interested in developing methodologies for the early detection of cancer through liquid biopsy analysis. But today, as I said, I will be discussing our work on detecting mutations in single cell datasets. So first of all, I mean, perhaps this is a not needed introduction, but why is it important to detect and to study mutations? So mutations of the genome are really the driving force of cancer as well as other diseases. And over the last years, we've learned that in our bodies, in histologically normal tissues, there are clonal expansions that in some instances drive various diseases, and perhaps clonal hematopoiesis of intermediate potential is really the best example. Detecting mutations is also important from a clinical perspective, as predictive biomarkers of drug response, such as mismatch repair deficiency, can be detected through the identification of specific patterns of mutations. And finally, from a more, let's say, basic science perspective, detecting mutations can be very useful, for example, in the study of development. And this is because somatic mutations accumulate as we age, and they can be used as genetic barcodes for lineage tracing. Then the question arises, okay, some mutation is important, but how can we detect somatic mutations? There has been a lot of work over the last decades, and so far mutations have been detected through sequencing of the genome, and this can be done in various ways. The most common one is bulk sequencing, in which the genome from a group of cells, usually many cells, is sequenced. And this has the limitation that only mutations present in most of the cells in the sample can be detected, due to the limitations of sequencing depth and the error rate of in this case, Illumina sequencing, which is a predominant technology. Sequencing mutations, detecting mutations in single cells is the most direct way to study mutagenesis, but current single cell genomics methods suffer from various biases and artifacts, such as dropouts, that really limit the application of these methods at the scale, although we very well know also a lot of work from Harvard Medical School, Boston Children's, etc., that there are really kind of exciting advancements in this area, such as the PTA method. And more recently, there has been an increasing interest in using duplex sequencing to really push the limit of detection to detect somatic mutations just present in a small fraction of the cells in a sample. But I guess the key point I want to make is that all these methods suffer from the limitation that by detecting mutations even in a single cell in a sample, we don't generally know in which cell types the mutations occur, and that doesn't apply to single cell genomics methods, but certainly applies to bulk sequencing and duplex sequencing. We cannot determine what are the rates of mutations across different cell types, and primarily, we cannot really understand what is the impact of those somatic mutations on cell differentiation, cell function, or normal genotypes. And finally, we cannot really determine which mutations are under positive or negative selection across different cell types. And really, these are the questions that we are interested in, right? So one thing is it's affecting the mutations, but the question that arises then is like, do these mutations matter? And there has been a lot of work in the cancer field, but more broadly, we don't understand for most mutations in cancer and in normal tissues what they are really how they are really affecting cell behavior. So to basically address this question, what we really need to do is to map phenotypes to genotypes, to really profile both the genotype and the phenotype, which can be gene expression or other type of, for example, the methylation or other phenotypes of the cells. And I would like to give you like an introduction to the method that we've developed by really telling you a bit about the story that we published a few years ago in collaboration with the Mulally and our Hormuz labs in Harvard Medical School. That was really kind of, it was really inspiring for us to develop this method. And also we learned a lot in terms of the possibilities of detecting mutations in single cell datasets. So in this project, we were interested in studying JAK2 mutant cells in myeloproliferative neoplasms, or MPNs, which is a type of blood cancer that develops through the ground expansion of a single cell after the acquisition of a JAK2 mutation. And here in this plot on the left, you see a UMAP representation of the major cell types that detected in these cancer patients. And as you can see, there is no clear cluster of cancer cells, right? So which means that the differentiation hierarchy in these patients is maintained, the hematopoietic differentiation hierarchy. So what has happened in the context of cancer research using single cell methods is that it is important to basically determine which of these cells are the cancer cells. And this could be done through multi-omic methods, right? So methods designed to profile both, for example, the transcriptome and detecting mutations in single cells. But these approaches usually are not scalable, right? And they are highly complex, they are not cost efficient, and in practice, they cannot be deployed at the scale. Other groups have relied on detecting copy number aberrations, for example, that can be detected in single cell RNA sequencing data based on the premise that increased copy number leads to higher transcription levels. But the problem is that in some cancer types, such as this one, where there are no copy number aberrations, we cannot use that approach, right? And certainly in normal tissues, that is generally the case as well. So what we did in this study was to basically genotype the single cell detection of the JAK2 mutation, as well as other mutations that we detected in whole genome sequencing data that we performed for these patients. And as you can see here on the right, we can see that most cancer cells, although they are detected across the entire differentiation hierarchy, are enriched in the erythrocyte and megakaryocyte and progenitor cells, right? So there is clearly a fate bias here that we could only determine through the gen profiling of the transcriptome and the genotypes. And to do so, again, this is the same plot. What we did was to detect the JAK2 mutation, which is the pathognomonic mutation in this cancer, through amplicon sequencing on the single cell library. So basically, one can perform amplicon sequencing on the same library that is subject to single cell RNA sequencing. And then we also perform amplification of mutations that were detected in the whole genome sequencing, but that were not JAK2. And you can see that here, the cells where we detected the wild type allele are colored in blue, and those where we detected the mutant allele are colored in red. And also, because we had whole genome sequencing for these patients, we tried to basically detect those mutations in the single cell RNA sequencing reach directly. And you can see that the cells with the mutation were also showed this bias. But what is more really important here that I want to highlight that will be relevant for the rest of the talk is that those mutations detected directly in the single cell RNA sequencing data allowed us to genotype hundreds of cells. And some mutations, such as one that we found in the intron of UPF1, was very, very informative for genotyping, for identifying the cancer cells. And this mutation was just a passenger mutation that we detected through whole genome sequencing. But then these results really prompted us to think, we cannot always do whole genome sequencing in all samples. And certainly for public datasets, we do not have much whole genome sequencing data. So could have we detected those mutations directly in the single cell RNA sequencing dataset without doing whole genome sequencing? Or in essence, can we detect reliably mutations in the single cell data? This is challenging because we all know that single cell datasets have a high error rate, which means that discriminating between mutations and artifacts is very hard. There are some challenges associated with technology, such as, for example, the fact that we only profile one allele per cell, which means that if we are not profiling the mutant allele, there is an inconclusive genotype. We can only say that we cannot see the mutation, which doesn't mean that the mutation is not there. And then there are other limitations, such as, for example, the uneven and low sequencing coverage across transcripts. However, there are some precedents in the sense that there are several methods have been published to detect, to basically genotype single cells through the detection of point mutations. And one of the kind of main algorithms in this space is Carderino, developed by colleagues here at the Sanger, which basically has in common with other algorithms that the first step is really the need for defining a phylogeny using somatic mutations detected with a reference sample. So, for example, exome or whole genome sequencing data. Once the somatic mutations have been detected in DNA-seq, then the algorithm basically genotypes the single-cell RNA sequencing data. And here, the key limitation is that there is a need for a matched reference sample that, as I said, we do not always have. And for most data sets that are out there, and we can think about the human cell atlas, for example, there is really no match sequencing data. So what we really think that is needed in the field, and that's why we developed the schematic, is that we really need an algorithm that should be well compatible with scalable and widely adopted technologies. So we don't want to really I mean, it wouldn't be very useful to have an algorithm that is only compatible with a fancy, sophisticated experimental protocol that no one else is using beyond a few labs, right? We really want to have an algorithm that works with the widely spread technologies, such as, for example, 10x genomics. We really want the algorithm to be highly specific, which means to have low false positive rates. Detecting mutation in single cell datasets is easy in a way because there are tons of variants there, but the problem is really removing the false positives, right? And those can be very detrimental for computational analysis, as I will show you later. And then what we really need is an algorithm capable of detecting mutations without having a reference DNA sequencing sample. And the main reason is that we really want to use the existing data sets, such as those generated in the context of the human cell atlas and other projects, as well as emerging data sets where there might not always be a match reference sample, such as, for example, such as H10. And that's basically, with all that background, we decided to develop schematic, which is a method that can detect somatic mutations. And by mutations here, I refer to single nucleotide variants, de novo in single cell profiling datasets. And by that, I'm referring to single cell RNA-seq and single cell ATAC-seq, as I will show you in a bit. So what are the principles, right? The tenets underpinning schematic. We consider that a late somatic mutation, and by late, we mean after organismal development, will only appear in, or should only be detected, right? If it's a true mutation, in cells that are descendants of the same progenitor and not across distant lineages. And it follows from the preceding that if we find a candidate mutation that is detected in multiple and distantly related cell types, so for example, such as a T cell and a muscle cell, those are going to be likely artifacts or germline polymorphisms, or perhaps embryonic mutations. So somatic mutations acquired very early, which can be sometimes, or in this context, misclassified as germline. And I will come back to this in the limitation section. Then we consider that only mutations with frequency higher than basically the background error rate of the technology that we are using can be detected, and be ensured that that's the case. And then, although it's not ideal, there are some sites in the genome that are error prone due to mapping errors, artifacts, or experimental issues. And in those positions, we cannot really detect mutations. And finally, we consider that there needs to be enough evidence to make a mutation call, right? So for example, we require that at least 2 single cells have a mutation. And here, I mean, sometimes this will decrease the sensitivity, but it's it's really not trying to maximize the specificity over sensitivity, just to have a highly accurate mutation call set. So these are basically the principles underpinning the algorithm. And let me walk you through how we've implemented it and how it works in practice. So the first step of the algorithm is to use to annotate basically cell types. And we can use, for example, the expression of marker genes when in the case of single cell RNA sequencing, which is the main application within or technology we've been working with. Once we have the annotations for every single cell, we split the sequencing alignment file, usually in binary alignment format or BAM format, into cell type specific BAM files, right? And that is represented here. So we split the sequencing rates into different files, and in each file, we only have the sequencing rates originating from cells of the same type. Next, we basically, for every position in the genome, we record the base count information. Sorry, so basically we record how many bases, how many reads support each of the bases, and we do this across the genome and for all cell types. And then using non-neoplastic samples, we determine the error rate. And to do that, we use a beta binomial. And this is important because then for every position and cell type, we determine whether the variant, the number of alternate reads is below or above the background error rate. So where there is a significant number of reads supporting the alternate allele at that position. And this is what we consider evidence for a mutation. And once we have identified those candidate mutations, we apply a set of filters to basically remove what are going to be likely not somatic mutations. And here what we use is a concept basically that has been used widely in the cancer genomics field, which is a panel of normals. And this is an approach where we analyze a large number of samples to identify those positions that are subject to recurrent errors or artifacts, where we cannot basically rely on to call mutations. We also remove positions in the genome in the case of RNA sequencing data that are in RNA editing databases. Also, we remove those candidate mutations where there is not enough support, right? Or where we find the alternate allele in other in cell types, in multiple cell types that are not closely related. And just to basically show you why it is important to, for example, model the background mutation rate, we've identified that there are some areas in the genome, in particular sign elements, where there is an enrichment of artifacts. So those are sites where we cannot reliably detect mutations, and those are sites that we remove. And here I'm showing you, for example, the distribution of the genomic distribution of those positions that we include in the panel of normals, and the enrichment of some of these elements with respect to the genome. So we can see, for example, that sign elements or areas categorized as low complexity and repeat masker are particularly enriched in sites that we cannot reliably call a mutation, and those are removed from during the calling process. Just very quickly, because I'm just going to discuss something that we've been asked about a lot over the last months, and is the question of how granular the cell type annotations need to be. So I would just start by saying that schematic can be used with different types of granularity in terms of these cell type annotations very easily. So it can be adapted. But the key point here is that we need to consider when a mutation happens or where it happens, basically across the differentiation hierarchy. So if we, for example, consider a mutation that happens late in a progenitor cell, and then we consider and we have 4 cell types, A, B, C, and D, and then we group them as AB is one cell type and C and D is another cell type, this mutation will be detected. And the reason is that the mutation will only appear in this group and not in the other, so it will be detected. And the same would happen to an early mutation here shown in red that happens in the progenitor cell common to cell type C and D. And in this case, we'll see that it will be in C and D. When we compare these two with A and B, it will be detected. However, if we consider each of these 4 cell types as independent cell types, right, A, B, C, and D, this mutation in green here, the late mutation will be detected because it will only be detected in one cell type. However, the red mutation, because it will be present in 2 cell types, it will not be detected by schematic, as it will be considered likely an artifact because it is present in multiple cell types. So depending on what type of mutations one wants to detect, the granularity of the cell types will need to be adjusted. And this is really something that is informed by the biological question one is interested in. In the following slides, and yeah, for mostly the rest of the talk, I'm just going to introduce you some of the analysis that we've been performed by applying somatic to various data sets. And this will allow me to show you some biology, as well as to show you why we are confident that this algorithm works. One of the things that or the three main data sets I'm going to focus on are these three corresponding to squamous cell carcinoma, ovarian cancer, and kidney cancer. And these are the ones that we've been using extensively for benchmarking purposes, because in these data sets, we have matched either exome or whole genome sequencing data, as well as single cell RNA sequencing. And in total, we detected 416 mutations in the DNA sequencing data that we use for benchmarking purposes. And just upfront, these are the results of the benchmarking where we compared the performance of the somatic, shown in blue here, against methods that are widely used for the detection of somatic mutations in DNA sequencing data, such as, for example, STRTA, as well as methods that have been specifically designed to detect mutations in single cell datasets, such as monovar and SC read counts. And what really stands out from this plot is that the blue bar, which is schematic in the middle, which corresponds to precision, is much significantly higher than the other methods. So while our sensitivity is sometimes lower than other methods, in these three cases, the precision is much higher, which means that most algorithms are calling tons of false positives that we are correctly classifying or not classifying as true mutations. And I will show you the data underlying these results moving forward. One of the analyses that we been relying on in this project is the detection of mutational signatures. And for those of you that might not be familiar with this concept, it really refers to the pattern of somatic mutations and mutational spectrum. And I will show you some examples in a second. But we have basically the mutations detected in the in this case whole exome sequencing data or whole genome sequencing data, and we can estimate the contribution of different mutational processes to the mutations that we observe. And we can see that the contribution of mutational signatures estimated using the mutation detected by schematic is very similar to the reference, which is the DNA sequencing data here in both the squamous cell carcinoma and the ovarian cancer datasets. But it is very different when we analyze data using basically you use the mutations detected by other algorithms. And something that was quite reassuring to see is that even for mutational signatures associated with a specific DNA repair deficiency, such as homologous repair deficiency here, we can basically identify such differences. And sorry, I know what's going on with my mouse. So in this plot, what I'm showing you is the distribution of the contribution of different mutational processes to ovarian cancer samples that are proficient for homologous repair, and those that are deficient for homologous repair. And as you can see, in those with homologous repair deficiency, the contribution of signature 3, which has been associated with this DNA repair deficiency, is significantly higher and is comparable to whole genome sequencing data. So really, the key point of this plot is that, or the summary is that we can detect mutational signatures, including those that are clinically relevant, using the mutations detected by schematic. Just to go a bit deeper into some of these data sets so you can see the underlying data. Here is, for example, the results for the squamous cell carcinoma. And you can see the burden of mutations computed as the number of mutations per megabase estimated for the tumor samples and matched normal skin samples. And we can see that these numbers are consistent with reports with the studies basically where sequencing of skin from normal skin and tumors has been performed. And also we can see that the contribution of the mutational signature associated with UV light, which is the cause of most mutations detected in the skin, both normal and tumor, is quite prevalent, which means that we can identify the mutational process underlying the mutation that we detect. And going back to the analysis of signatures, we can see here the mutational spectra corresponding to the mutations detected using a schematic in the tumor and the normal skin, and the similarity is very high, corresponding to the UV light mutational spectrum. How does this how do this analysis basically look like when we use other algorithms that are not schematic? So here you have at the bottom the mutational spectrum for those mutations detected in the cancer samples using the exome sequencing data. The second from the bottom is the spectrum corresponding to the mutation detected with the schematic, and you have here the similarity with the exome sequencing data, which is very, very high. One is the maximum here, the cosine similarity of this spectrum. And you can see that for the other algorithms, the similarity is really much lower, both in quantitative terms of cosine similarity, but also when we look at the spectra. You can see that these two are matched very closely, whereas the others are really different. Again, showing that the mutation detected by a schematic are true, and the schematic is highly specific as opposed to the other algorithms. One of the other analyses that we were interested in is computing the burden of mutations. So really the then estimated that as the number of mutations per megabase, and we can compare the results between the exome sequencing data here on the x-axis and the rate estimated in single cell RNA sequencing data. And we can see that the correlation is very high, which means that we can estimate mutation rates with schematic, which is important for some analyses, such as for example, the rate of accumulation of mutations in healthy tissues as well. And I will come back to that in a second. One of the data sets that we've been analyzing and using a lot for development of schematic as well as for discovery is a colorectal cancer data set. That primarily comes from one publication, but we also use data from a second one, which is indicated here. And this is interesting for the following reason. In colorectal cancer, we have tumors that are considered microsatellite stable, and those that have mismatch repair deficiency or microsatellite instability, which have a high mutational burden. And this is very useful for development purposes because we know that in MSI samples, which are colored here in red, the rate of mutations is much higher than in those with microsatellite stable phenotypes, which are shown here in blue. And in these cases, we also have matched samples from the colon, in which we know that the mutation rate is much, much lower. So in essence, what I'm showing you here is the number of mutations per megabase, or its bar here represents a tumor sample on the left, and on the right, we have the matched normal colon samples. And we can see that those samples that are microsatellite unstable have a much higher mutational burden as compared to MSS tumors as well as the normal samples. And these results are consistent with the rates that we estimated previously in TCGA samples for, in microsatellite unstable MSI samples from TCGA. And you can see the rate of somatic point mutations per megabase, which is comparable to what we estimate using the single cell data. If we look a bit deeper into this data set, we basically can determine the mutational processes operative in these cancers. And what we determined is that in the MSI samples, we did detect a strong contribution of mutational signatures associated with mismatch repair deficiency. And in the colorectal cancers with microsatellite stability, we did detect the strongest contribution was assigned to clock-like mutational processes that are reflected here or indicated by the single base substitution 5 and 14. We also, which is related to what I've mentioned, the burden of somatic mutations, the rate of somatic mutations that we detect in the single cell data for microsatellite and stable tumors is comparable to what we determined using exome sequencing data, again reinforcing the notion that with schematic and single cell datasets we can estimate mutational burdens. And something very interesting in this plot that we detected is that the tumor with the highest burden of somatic mutations was classified in in the paper reporting the data as microsatellite stable, but how could it be if the mutation is mutational burden is so high, right? One would expect that this is a misannotation and this is indeed a microsatellite unstable tumor. But when we perform mutational signature analysis, we realize that the high number of mutations in this tumor is not driven by microsatellite instability or mutations in the mismatch repair pathway, but rather by mutations in poly. And when we plot basically the distribution or the mutational spectrum for the sample, which is here at the bottom, we can see that it closely resembles the mutational signature associated with polydeficiency as reported by the COSMIC database. You can see here. So this means that really somatic can detect clinically relevant mutational processes in single cell RNA sequencing datasets, even for a single sample. And coming back again to the comparison against other algorithms, in this dataset we have samples with either microsatellite instability or polydeficiency, which are characterized by a very high mutational burden. Samples, tumors that are microsatellite stable, and therefore the mutation rate is much lower, and much normal samples shown in gray here, where the mutational burden should be very low. And here we can see on the y-axis the number of mutations per megabase across, for these two groups across different algorithms. And you can see the 5 algorithms against which we compared somatic assign basically a comparable mutational burden to the 3 groups. And by contrast, we can see here on the right that the somatic correctly identifies that the mutation rate in microsatellite unstable tumors is much higher as compared to MSS tumors, and that these tumors have a higher mutational burden as compared to matched colon samples. Again, highlighting the high specificity of the mutation detected by somatic. And then this data set, jumping back again to basically tuning the parameter and development of somatic. One of the key questions that we had throughout the project is how many cells do we require to have the same mutation to make sure that the mutation is real? And I've said at the beginning that we require at least 2 cells of the same type to have a mutation to make a mutation call. And the reason why we basically selected that cutoff is based on this analysis. The colorectal cancer dataset is very useful for development because as the samples are high, part of the samples, the MSI ones are hypermutated, we have a large number of mutations to do analysis with. And what we did in this precise analysis is to perform mutational signature analysis on the mutations detected in only 2 cells, cancer cells, epithelial cells, 3 cells, 4 cells, 5 cells, etc. And then we compared the assignments or the contribution of different mutational processes to the spectrum, right, that we detect. And as you can see, the results are very consistent, even when we require only 2 cells to have the somatic mutation. So the results gave us confidence to basically say that when we have at least 2 cells with the mutation, we can trust the call, in addition to the other filters. But this is kind of using the data or tuning the parameters of the algorithm in a data-driven way. So I guess detecting mutations in cancer might be easier because we know that the clarity of most mutations is going to be high. So next, we wanted to assess whether somatic can detect mutations in normal samples, and we started by reanalyzing the data set from the MPM patients that I have presented at the beginning. And what we can very briefly, it's the mutational signatures that we detected are consistent with the mutational signature analysis that we performed on matched whole genome sequencing data that we had for these patients. And we could also recapitulate the relationship between the mutational load and the age of the patient at diagnosis, which we know there should be a linear relationship, and that's something that we recapitulated in this analysis, suggesting that even in samples with a low mutational burden, we can confidently detect somatic mutations. We next were very lucky to working on in this project when the GTEx analysis, the single cell GTEx dataset came out, and this is the next single cell RNA sequencing data from various individuals and from multiple tissues. And by applying schematic, we could identify that the mutational signatures that we detected in the mutations called by schematic were consistent with studies, DNA sequencing studies of normal samples, primarily. So the mutations we detected were primarily explained by signatures 5 and 40. And this allowed us also to estimate the number of mutations per cell across various cell types and across various tissues. So basically, we estimated the average number of mutations per cell across diverse cell types in the human body. And we also did that using a single cell ataxic data set released in 2021, where we, in this case, is DNA sequencing because ataxic data. And again, the mutations that we detected were primarily explained by mutational signature 5 and 40, consistent with DNA sequencing studies. And this allowed us to basically extend our catalog of mutational rates across more tissues and more data sets. At this point, we were a bit worried, right, that there might be some batch effects and technology-specific issues that might affect the rates of mutations per cell type that we estimate. So what we did was basically look for cell types and organs that have been profiled across different studies, and we focused on the heart because we could find the heart cell as the data set generated by the Sanger, where single cell RNA sequencing was performed on a large number of samples, heart samples. The heart was also profiled in the GTEx study, and as well as in the ataxic study. And here, what I'm showing you in this figure is basically the estimated number of mutations per cell and per genome, using the mutations detected by schematic in the single cell RNA sequencing data from the heart cell atlas study, in the GTEx study, and in the ataxic study. And the samples are stratified by age. And as you can see broadly, the mutation rates that we estimate are largely consistent, indicating that the schematic can really detect accurately or estimate mutation rates across different profiling technologies and across datasets. And to get further confidence in these results, at the time of when we were just basically wrapping up this manuscript, there was a very nice publication by Alice Lee and Christopher Walls on single cell whole genome sequencing of human cardiomyocytes, and we used the mutation rate estimates from that study to compare them against what we could estimate using schematic, and we can see that there is no significant difference in the rates that we estimate. And I would like to emphasize that cardiomyocytes can be polyploid, and we don't have that information for the single cell RNA sequencing data. So these results are, inherently noisy, but still we could really estimate quite accurately the mutation rate per cell for cardiomyocytes. Next, as we can detect the somatic mutations across different cell types and tissues, we wonder whether we could study the patterns of clonality, and this was inspired by this nice landscape study by the Sanger, where 29 histologically different structures were sequenced using the laser capture microdissection approach that they have developed. And what you can see here is that, or the point I want to make is that there are some tissues that are or histological structures that are highly clonal, such as, for example, colonnade crypts, and this is reflected by a peak of the variant fraction of mutations at around 0.5. But there are some other tissues where the clonality of mutation is much lower, and we say that those tissues are polyclonal. So we wanted to assess whether we could estimate the patterns of clonality of the mutations across various tissues using the mutations detected by schematic. And to do so, we first basically computed whether the buff variant tail fraction of the mutation detected in exome sequencing data for the squamous cell carcinoma dataset correlated with the fraction of cells in which we detected a given mutation in the single cell RNA sequencing data. And you can see that there is a positive and significant correlation. And I would like to emphasize that obviously cancers are highly polyclonal, so that and that the same samples didn't undergo exome sequencing and single cell RNA sequencing, so we can expect some noise here in this data as well. But still, we see the positive correlation. And here I'm reporting you, I'm showing you, sorry, the fraction of cancer cells with that have a given mutation. And you can see that for the matched normal skin samples, there is a peak at very low cancer fraction values. And for the tumor samples, we see a peak at 0.5 for the clonal mutations, and then a subclonal peak. And we know from the exome sequencing data, these tumors are highly heterogeneous and that they have like a small number of clonal mutations. However, if we look at the cancer cell fraction, again, the fraction of cancer cells in the colorectal tumors that have a mutation, we can clearly see here both for the hypermutated polydeficient and MSI tumors, as well as the MSS colorectal tumors, we can clearly see a peak of cancer fraction at 0.5 corresponding to the clonal mutations, and a peak at lower values corresponding to the subclonal mutations. However, when we look at non-epithelial, non-cancer cell types from the tumors or the matched normal samples, we can see that the peak in distribution is below 0.02, and this is really corresponding to subclonal mutations, or meaning that most of the mutations we detect are only present in a subset of the cells in the sample. And when we look at, and just to show you an example, at the distribution of cancer cell fraction or the mutant cell fraction for the different cell types in the hard cell atlas, we can see that most of the values are below 0.2, again indicating that most of the mutations that we detect are subclonal, or in other words, that somatic is capable of detecting somatic mutations in polyclonal tissues. And finally, one of the applications that we were very interested in is in studying clonal heterogeneity in cancer. We'll note that clonal heterogeneity is a hallmark of cancer and underpins, for example, drug resistance. But when much of the work that has been done in understanding clonal heterogeneity in cancer has relied on identifying which mutations are clonal and which are subclonal based on the variant tail fraction distribution that can be detected in bulk DNA sequencing data. But the problem is that we cannot assign mutations to subclones using bulk sequencing data. So here we wonder, can we do so, right? Can we find patterns of mutations that coexist in the same clones? And can we identify some structure in the single cell RNA sequencing datasets from cancers to then assess phenotypic differences between clones? And to do this analysis, we use data generated by the Shah lab at MSK for ovarian cancers. And this data is particularly suitable for this analysis because we have site-matched whole genome sequencing as well as single-cell RNA sequencing, which also allows us to compare the cancer cell fraction of the mutations in the whole genome as well in the single-cell RNA-seq. And this is just an example of a tumor spectrum 03. And in this heat map, what I'm showing you here is on the y-axis, a set of somatic mutations detected by somatic, and each column here represents a single cell. The colors in this color bar represent the anatomical, the sampling site of the tumor where they were sourced, and then there is this unbiased clustering based on the mutations. Red means that we could detect a mutation. When we didn't detect the mutation, but we did detect the wild type allele, we have white, and gray refers to those two mutations that could not be genotype in the cells in a given cell because of primary lack of coverage. So as you can see, for many mutation single cell pairs, we don't have information, and that's something I'll come back later. And what stands out from this analysis is that there is a cluster here, which of cells that primarily correspond to or have been sampled from the right upper quadrant that are defined by the presence of these mutations. So now, if we zoom in this area, that's there is on the right hand side, we can see that the cells from this tumor region, the right upper quadrant, can be divided into two major clones that are defined by the mutations here indicated on the y-axis. And what is really exciting about this analysis is that we could identify, or these two clones correspond to basically clones defined using copy number information. So we basically ran a NumPad algorithm developed by the Karchenko lab at Harvard Medical School on this analysis to group the cells based on copy number information. And we can see that the clonal assignments based on copy number profiles align very, very well with the clonal assignments made based on the mutations detected by a schematic, meaning that we can identify these subclonal populations in cancer datasets directly using the schematic. And something that's also very exciting in this analysis is that we can see that some of the mutations were detected in the whole genome sequencing data. This key map here represents the cancer cell fraction in the whole genome sequencing data for the mutations here on the y-axis. And some of them were really detectable in the whole genome data, but some of them were there wasn't basically no evidence for those mutations in the whole genome sequencing data, as they were below the limit of detection, but they were very nicely detected with a schematic. And then when we basically map those mutations on the UMAP to show transcriptional differences, we can see that the clones estimated using the schematic align very nicely as well with the clones detected by NumPad based on copy number information. So I've basically highlighted some of the nice and interesting analysis that we can do using a schematic, but there are certainly limitations to this tool, and that I would like to highlight. The first one is that we really need two cells to have a mutation to be confident that the mutation is real, and this means that mutations present in a single cell cannot be detected, which means that some mutations will never be detected if just present in one cell, and some mutations might not be present, might not be detectable by a schematic if there are downsampling issues. So for example, if a small number of cells is sequenced. Embryonic mutations, by that I mean those mutations happening very, very early within development, are very hard to detect at this point in most datasets, because they are as embryonic mutations will appear in multiple tissues, the schematic will think that they are likely going to be germline polymorphisms. So if you are interested in detecting embryonic mutations, some tweaks with the cell type annotations might be needed. And also it would be needed to sample cell types from highly distant lineages to detect them. As you've seen in the ovarian cancer example that I've just discussed, where we've done the clonal reconstruction, we cannot genotype many of the mutations in a given single cell. So this means that the mutation by cell matrix is really sparse, and this is just a limitation of the low sequencing depth of single cell datasets. It's nothing we can really do much about, and it's also related to expression levels, etc. But that's for some applications, and in some cases, it's certainly going to be a limitation. Also, the sensitivity for detecting mutations, it really depends on the technology, right? So we've primarily focused on 10x datasets where there is a clear bias towards either the 5' or 3' end, although we can also detect intronic mutations, etc. But there is clearly an uneven distribution of coverage across the transcriptome, which means that the sensitivity for mutation detection is also affected by that. And finally, we really rely on accurate cell type annotations, and that requires hard work. And sometimes that is not possible, or for some public datasets, annotations might not be reliable. So this is something that is not necessarily a limitation of the algorithm, but it really requires careful thinking and consideration when running the tool. We're very excited about this line of work, and certainly we are already working on future directions. And the first of which is extending Schematic to be compatible with long-read sequencing datasets. And for example, PacBio recently co-developed, I think, with the next an assay called MASSIC, which plans to harness the long-read sequencing capabilities of high-five rates and combine them with 10x data. And certainly this is very exciting because the error rate of high-five rates is very low, and increasing the breadth of coverage will increase the sensitivity for mutation detection. And if at some point we could read out entire transcripts, we would be in a position to study positive selection or negative selection at cell type resolution, which is something I'm particularly interested in. We've been primarily applying Schematic in the context of methods development, but we would be very happy to collaborate and work in applications related to lineage tracing. I think there is a lot of potential there, not perhaps to reconstruct high resolution lineage trees, but to assign, to basically ascertain which progenitors, which are the progenitors of a specific cell type, etc. That is something that could be analyzed using Schematic. Another area where I think the combination of cell-phenotypic information as well as lineage information can be very, very valuable is the study of cell plasticity in cancer. And that's an area we're also very interested in. And finally, we've been focusing on human data sets due to primarily data availability, but Schematic could also be used to study somatic mutations beyond humans in other contexts where there is a good reference genome. But I think now with the increasing availability of high quality reference genomes for other organisms, that is certainly within reach. And so in terms of conclusions, I would like to highlight that so the Schematic is a tool that permits to detect somatic mutations in single cell datasets without requiring a matched reference sample. And this is really one of the key features that sets Schematic apart from other tools. We've shown that Schematic has high specificity, much higher than existing methods. So whenever this is important, which is in most of the on most occasions, Schematic is going to be advantageous as well. And it's not only that we can detect mutations, but we can use the mutation that we detect to estimate mutation burdens very accurately, as well as the detection of mutational signatures, including those of clinical relevance, such as mismatch repair deficiency or homologous recombination deficiency, as I've shown. And finally, really, Schematic opens the possibility to map genotypes to transcriptomes to study clonal heterogeneity in the context of cancer and somatic evolution in normal tissues. And there are very exciting initiatives starting now, where clonal mosaicism across the body is going to be the key focus of a study. So we really hope that Schematic will be of use in those contexts. Finally, I would really like to thank my entire team, but primarily Fran, who's been the key developer of Schematic and has been working on this very hard, as well as our collaborators, primarily Sahand and Anne, with whom we worked on the NPM project, and our funding. And thanks you for your attention. I would be very, I'm very excited to take any questions you might have and discuss further about Schematic and these areas of research. Thank you very much.
Thank you so much. And a very great talk. I think we have one question from the Q&A. So I'm not sure whether you can read the question or not. Yeah. If not, I can read that and you can answer that.
Yeah, I can read. Yeah, thank you. So the question is related to whether we could apply a Schematic on vision data using basically spot level data and how we should interpret it. And this is related to the fact that, for example, InferCNV, a tool to detect copy number aberrations in single cell data, has been used in vision data. We haven't tried ourselves because we don't have much vision data. In principle, it should be possible, but interpretation should be slightly different. We could in the same way that now we annotate cells, cell types, right? And we say, okay, this mutation is in this cell type and not in this other. With the spatial data, the concept of cell type will be basically, or should be interpreted as regions, right? So we could detect which mutations are in a particular region and not in others, unless we had single cell resolution, which with the current vision platform we don't have. So we can apply the tool, but the type of mutations that we can detect will be certainly different.
Great. We have another question from Michael. I can read the question. So two questions. Well, I guess two questions. What is the coverage per base per cell in the analyzed data set? And how many wild type reads do you need before you consider a cell negative for a mutation?
Yeah, thank you very much. So in terms of the coverage per base per cell is at most 1. It's either 0, no coverage, or 1 read. Sometimes we can find 2 or 3, but that's really not the, I mean, in the vast majority of cases, we have only 1 read per cell per position. And that is why whenever we detect a wild type allele, we don't know if we are sampling the wild type allele and not the mutant allele, or that cell has 2 wild type alleles, right? So that's why I mentioned that sometimes we can have inconclusive genotypes, because if we only see the wild type allele, we don't know if we are simply not sampling the mutant allele. And in terms of the number of wild type reads that we need to consider a cell negative for a mutation, we are so let me think if I understand this correctly. So when we have we need at least 2 cells of the same type with a mutation. That's a requirement, and then we require at least I don't remember the exact numbers, but we require that the mutation is not present in other cell types, or at least above the level of significance for the background mutation rate. And in terms of if we are interested in a particular cell to consider that cell is negative for the mutation, we will need to sample both alleles. And as I've just said, that rarely happens. And even if we had 2 reads, we would not know if they come from the same allele or just from one, unless we could face with a nearby heterozygous SNP, which in our experience is going to be very hard in single cell RNA sequencing datasets. Hopefully that answers the question. Otherwise, please let me know.
Great. Do we have any other question? If you have any more question, put it either type in the Q&A or just raise your hand. We can also unmute you. Okay. Sanjita.
Hi, Isidro. It was a wonderful talk, wonderful data, I'm very optimistic that this could be used. And I'm so glad that you found, my paper to, model. I'm very glad. And I have one question and one probably comments that, what you saw that there was some variability between your call and our analysis. It could be due to that cardiomyocytes are not, they are actually post-mitotic cell types. So I was surprised. I thought that there are few clonal mutation, but mostly they were non-clonal from our study, what we found. So that's why it could be, that, there was lower. And then my question was that because you need at least 2 mutations, so this, so the 2 cells have to have the same mutation, is that correct?
Yes.
To call, right? Okay, got it. So then, it will be difficult to use this in a post-mitotic organ where you don't see, more clonality. Is that correct?
Exactly, yeah. In highly polyclonal tissues, post-mitotic, such as the heart or the brain, right? And the mutations we'll be able to detect will be either those that happen, before during development, late in development, right? Or otherwise it's going to be very hard. And there is also the problem of sampling, right? In some datasets, we only have a few tens of cells of a given type, and that is going to be undersampled. Even if the mutation was present in multiple cell types, that would also limit the detection for mutation.
Great
but yeah,
work.
thank you. Those are good points. Yeah. Thank you.
Great. Well, I don't think we have any more questions. Well, thank you so much for the great talk.
Biju had a question.
Biju? Oh, yeah. Biju, I think you can.
I have a quick question. Thanks, Isidro, for the wonderful talk. So it's about the input technology. So I'm guessing that you're using full length transcript for this. So are you using 10x technology for your input? Because most of the data that comes from there is from 3' or N libraries. So are you going to use the 10x data for your mutation analysis? That's what my question is.
Yeah, so all the single cell RNA sequencing data or results I've discussed today is 10x data.
So then how do you calculate mutation rates from sequences that are not covering the entire gene? That's my
Yeah, of course. So 2 points. So first of all, although we're going to have an enrichment of reads at the 3' end, for example, in the 3' end datasets, we know, and for example, work on RNA velocity, Peter Karchenko and others, we know that some reads are going to map to intronic regions, right? So there is going to be also a bit of coverage across the transcript, and that can be very useful, something that I have not discussed, because one can detect mutations in intronic regions that can be useful for signature analysis or also for marking subclones. That's one thing. And concerning your second question, to estimate the mutation rate, we just extrapolate, right? So if we have coverage in X many bases and we detect Y many mutations, we just extrapolate across the entire genome. And because we only detect, usually we can only profile one allele per position, we estimate the mutation rate per haploid genome. That's why in the paper we discuss rates per haploid genome. And obviously, one of the limitations there is that if the distribution of mutations is different across the genome, and we know that that's the case, and we're detecting mutations in regions that are enriched or depleted for mutations, our estimates are going to be not as accurate, right? But that's something that we've shown that they are accurate enough, and perhaps we think it's because we in the single channel sequencing data we sample both exonic and intronic regions, but that is certainly a limitation. So with the full transcript datasets, which we don't have, and hopefully we'll have soon, we should be able to perhaps increase the accuracy of those estimates. But as of now, I cannot comment more on that because we don't have data to compare.
Okay, thank you.
Thank you.
Great. I think there's not a questions. Great talk. Thank you so much. I don't think we have any more questions now.
Okay, well, thank you very much, everyone, for attention and for the invitation. It was a real pleasure. And if you have any other questions or comments or do want to discuss further, please reach out. I will be delighted to talk more.
Great. Have a nice day.
You too. Thank you.
Thank you, Sajita.
Bye-bye.
Awesome. Yeah, bye.